Hello and welcome back to the second part of this week's material on biotechnology. And we're going to be focusing on what we've all been living through, this COVID pandemic, and making some connections with the material in class. So we're going to be talking a little bit about PCR, which we've now been introduced to in last video. And we're going to talk about the test. We've been talking about mRNA, and we're going to see how that relates to the vaccine. We've been talking about nucleotides and those A's, T's, C's, and G's in DNA, and A's, C's, G's, and U's in RNA. And we're going to talk about one of the only drugs, therapies that's been approved, and also the role of proteins, which we're going to see as antibodies. So I've got a whole bunch of different links, as I'm sure you are all well aware. There's a lot of information going out on this COVID pandemic. Some fantastic, like this article from Scientific America with some fantastic visuals which make you see the virus right here and these different components where we've got the genetic material inside right here. It's encased in these lipids and proteins. Okay, so what does that really mean? What are we going to be talking about? Obviously, we're going to be talking about COVID-19, which stands for coronavirus disease from 2019, caused by the SARS-CoV-2, which is the SARS coronavirus, and it's the second SARS virus that we've isolated as a human pathogen. And what does it mean to be a coronavirus? It means that it's a positive strand RNA virus. So what positive strand means is that this RNA that's in the inside, that's the genetic material, that is what specifically is injected into the cell to make it infectious, is just like mRNA. So it's positive sensed, meaning just like we saw for gene expression, where we go from DNA to RNA to protein, it's positive sensed, meaning that it's just like mRNA. So that the strand itself, once you get that viral genome into a host cell, it is able to instantly be made into proteins. We're instantly able to translate those viral proteins. Now, being a virus, it's been called the bad news wrapped in a protein, because of course what a virus is going to do to enter a host cell, and it's going to produce those viral proteins to make up the virus, and it's going to replicate its genome. So as we just said, its genome for this SARS-CoV-2 virus is RNA. So just like we saw for DNA, we need to copy that DNA in order for our cells to reproduce. In order to make more virus, that virus needs to copy its genome. It needs to copy its RNA. So one of the proteins that this virus makes is an RNA polymerase that uses RNA as the template. So this is something that's fundamentally different from how our cells operate, going from DNA to RNA to protein, where it's the DNA gets replicated and uses DNA polymerase. This is going to be an RNA template RNA polymerase. And so what that RNA polymerase is going to do is going to copy that viral genome. So one of the first treatments that has been approved in the United States by the CDC, although it's not actually recommended by the WHO, is what we call remdesivir. What it is it's a nucleoside analog. And what that means as a nucleoside analog, it means that it looks just like a nucleotide, but it's not able to have something added after it. So the drug remdesivir, which has been approved, looks like this when it's injected, and then gets modified within the body by basic enzymes that are going to cut it and phosphorylate it and end up producing this nucleotide triphosphate active metabolite, which can be utilized by that RNA polymerase, but it cannot be added to, just like we have the issue with that 3' end. You can't actually keep adding new nucleotides to. So this drug is going to inhibit the RNA polymerase. And we have seen that it reduces recovery time slightly, so it has been an improved drug, but it is not a miracle treatment that is able to completely eliminate that viral replication. So some other information you may have previously heard about, and I just mentioned earlier, is that COVID-19, caused by that SARS-CoV-2 virus, isn't the first coronavirus. In fact, I can pretty much guarantee you have all been infected by a coronavirus previously, as there's 4 strains that make up the common cold. And this current pandemic isn't even the first epidemic. Back in 2003, the SARS-CoV-1 virus did cause an epidemic, and hundreds of people died. But one of the characteristics of that virus that ended up being fortuitous was that people weren't infectious until they were symptomatic. People started to feel bad at the same time there was enough virus in their body to actually be able to spread to another individual. And this is quite different than what we see with the SARS-CoV-2 virus, where we see there's a huge variation in immune responses. There are individuals that are completely asymptomatic and have absolutely no idea that they have this virus replicating within their airway epithelium and being produced by their body and being released as they breathe, or in fact, go to the bathroom. So there's people that are completely asymptomatic, and then tragically, there are other individuals who end up dying from the disease. So that is why it is so super duper important for us as a country and as a world to start to be able to test to see who is actually infected with this virus. So there's a few different ways we can test to see for active virus. One of them is what you learned about already. So this PCR test. So this PCR test, as you learned earlier, it's amplifying DNA. In the case of the COVID tests, you take a swab of the nasal cavity, essentially, and you can use primers to see, is there any of that viral genome present? And you can actually look to see quantitatively how much virus is present based on how many cycles does it take to reach a certain threshold. So again, what's happening in PCR is you're getting these temperature changes and you're getting these rounds of replication. And so if you start with more virus, you are going to have more virus. You're going to have more of these particular sections of DNA that can be seen earlier in that cycle. So you can use PCR, and that's sort of the gold standard now for determining for active infection. But there's also what we call these antigen tests. Now these antigen tests aren't looking for viral DNA, they're looking for viral proteins, where you have these lateral flow, essentially pieces of paper, you can think about it, that have been coated with different antibodies. So we're going to talk more about antibodies in a second, but these are proteins with a specific shape that are going to recognize a very specific molecule, and you are going to be adding your sample, and it's going to move across that strip, and if the viral protein is present, you will end up getting a line that you can see based on these other antibodies that have been added to the actual strip. So these antigen tests are quite quick. These antigen tests can be done in just a few minutes by anyone, essentially. Whereas these PCR tests, you need to have a PCR machine that can go through those different cycles and temperatures. And even though they actually have been shrunk in time in terms of being able to be done in minutes versus hours, this still isn't something that you're going to be able to do in your home. Whereas these tests, just like pregnancy tests, anyone can actually do. When we're thinking about testing, we can look at when these tests would actually be positive, and thinking about the range in human responses for what we see and the range what we see in immune responses, where that PCR test is going to be one of the earliest tests to see once you have enough virus that you can actually get just a few copies of that virus into the test, you can start to see it. And as long as you've got viral genomes, you'll be able to see it. So you can see it quite a while. Whereas those antigen tests, they're only going to be present if you have enough of that protein, so it's a much smaller window. And then we also see over here that there's these antibody tests. So these antibody tests for IgM and IgG aren't looking to see if there's virus present. They're looking to see, did the immune system generate a response? So when we say an immune response, that's the same thing as we're trying to get out of a vaccine. And again, this isn't 205, where you'll learn all about the immune cells, so we'll just keep it at the most simple, that we've sort of have two different big parts of our adaptive immune response, where we've got B cells that make antibodies, and we've got these T cells, which go and actually remove and kill the infected cells, and there's different types that also help initiate the immune response. But we are currently also doing antibody tests to see if people have these antibodies present in their bodies. So the test is sort of similar to here. It's an ELISA test where we're trying to see if our body is producing the actual antibodies. So this is an ELISA test similar to what is done for an AIDS test. And why do we care so much about antibodies? Because antibodies do two really important things. So we can think back to that idea of the host cell and the host cell plasma membrane with all those different proteins and those different glycoproteins. So it's these glycoproteins which are what viruses actually attach to. So this is how this viral genome is going to enter this cell. It is first needs to attach to a protein, and then through that lipid membrane it'll be able to fuse. So what antibodies do, antibodies are specific, they're proteins, so they have a specific shape, and they are going to bind to that viral particle by the same protein that is going to bind to that host cell. So what antibodies do is they block a virus from being able to enter and infect a cell. What antibodies also do, you can kind of think of antibodies like a sugar coating that makes something so much tastier for the immune cells. So in addition to physically blocking virus from entering a cell and causing an infection, antibodies will also cause those viral particles to be eaten by the immune system more rapidly. So the production of antibodies is really important for what we're trying to initiate in a vaccine, but it's also a treatment option by taking convalescent serum. So what is convalescent serum? From an individual who has recovered from the infection, so whose immune system has generated antibodies, to isolate that part of the blood and to be able to give just those antibodies from a previously infected individual. That's what convalescent serum is. And it's not just utilized in COVID-19 infections, but rather for multiple different types of infections for over 100 years. What is new and was just approved is actually what we call monoclonal antibodies. So monoclonal antibodies, rather than getting all the different antibodies produced from convalescent serum, is taking particular cells from a mouse and fusing them, creating these hybridomas, which will produce large quantities of antibodies. So then you can just give someone just that purified antibody that we know will block that viral attachment. So these treatments are definitely very exciting, but what we've all really been waiting for is a vaccine to generate that immune response so that someone is no longer susceptible to infection, not from getting the virus, but from getting the vaccine. So all previous vaccines that have been released are either live attenuated virus, where it doesn't cause disease, but rather just generates that immune response, or dead viruses, which are incapable of reproducing, but still generate those B cells and T cells, or just taking parts of the virus. So a dead virus would be the whole virus, but other vaccines just use a protein or just use some of the lipids from it to generate a response. Now, this new vaccine that we're all getting excited about should sound vaguely familiar because it's what's called an RNA vaccine. And we spent a bunch of time talking about RNA. RNA is what is produced from transcription and what it is used in translation to make a protein. And this is a totally new type of vaccine where instead of injecting, again, whole virus or just proteins, you're injecting this RNA. And so this is mRNA, and this is, again, positive stranded RNA, when it's able to enter a host cell, and particularly a dendritic cell, so particularly these cells of the immune system that are responsible for activating those B cells and T cells, it is able to be immediately expressed into that protein of interest. So these messenger RNA vaccines that we've been hearing about quite recently is a whole new technology using this new molecule of life, using these nucleotides to generate an immune response. And what we can see here again is just how important it is that this RNA has to be protected in a way so that it's not going to be instantly degraded by our host cells, and that it is able to enter these dendritic cells and be expressed to make that protein of interest to activate the immune cells. So there's a lot to be hopeful about and a lot to be excited about. But until then, I cannot stress enough the importance of wearing a mask and practicing social distancing. So I've linked to 2 amazing articles that show you some fantastic graphics for how viruses are spread and what we can do with masking to try to reduce viral spread. So stay safe, and I'm very excited to check out our discussion for this week. Be well.